So in this video, we will be discussing carbohydrates, one of those four categories of macromolecules. Okay, so just in general, we do have different chemical names for different categories within carbohydrates. So there are, like, to call something a sugar is a chemical name, that tells us it's a maltose or fructose or lactose, and the same thing when you're calling something a starch. But they're not necessarily interchangeable with carbohydrate. Carbohydrate is the larger category. These macromolecules are made of carbon, hydrogen, and oxygen, and that's it. Okay, just three atoms, and they all are in the monomers. They all come in a 1:2:1 ratio. So for every one carbon, you'll have two hydrogens and one oxygen. So it's real easy to figure out their chemical formula. So, for example, glucose is C6H12O6. In fact, all of our 6-carbon sugars are exactly that. Now, monomer is a generic term that can apply to any of the macromolecules, but specific to carbohydrates is the word monosaccharide. So it has the same beginning. Mono means one, and then saccharide is another term for a sugar. So monosaccharides. And you can have different sugars that have differing amounts of carbon. Usually the ones that we focus on in this course are going to be the 6-carbon sugars. So we'll talk about glucose, galactose, fructose. But we do also talk about the 5-carbon sugars, ribose and deoxyribose. Those come up in DNA and RNA. But chemically, there are many different kinds of sugars. And they are, various ones are present in the body, but we're not going to focus on those in this course. If you look up close, you can see the sugars. This is their chain form. When they're in water and living systems, they actually form a ring. So we actually don't, as we go through the course, you won't actually see sugars looking like this. Okay. So the monosaccharides, and also you can have a disaccharide when you have two monosaccharides that bond together covalently. These are basically what living organisms use to extract chemical energy. We use the hexose sugars, so they have 6 carbons, and you do need to know these 3 monosaccharides by name, and you need to know about them. And you also need to know these 3 disaccharides by name and about them. So glucose, galactose, and fructose. And because they form a ring structure in water, they kind of look like hexagons. So that's how we draw them. Typically, as a shorthand, we'll just draw the hexagon and leave out the other atoms. Okay, but glucose is our main sugar that organisms use for making ATP. What we call, like, as a shorthand, if you've ever heard the term blood sugar, like that's the main sugar in your blood. Galactose is an isomer of glucose, and it's very similar, and it's also used for making ATP. Now, fructose is made by plants as a part of photosynthesis, and fructose can be used by our body to make ATP. But it shouldn't make up a huge part of our diet, and that's why you hear campaigns, trying to get us not to be using so much high fructose corn syrup. It's easier to digest than sucrose, and it tastes very sweet, so it's easy to consume too much sugar by having that high fructose corn syrup. So if you combine various combinations of these, you'll get the disaccharide. So if you have 2 glucose molecules that bond together via dehydration synthesis, then you'll wind up with a disaccharide called maltose. Sometimes this is called malt sugar. You may have come across it if you've ever had the soda Malta, or if you've had a malted milk ball, like a Whopper, or if you have heard the term malt liquor. Okay. It is commonly found in seeds. So in seeds, they store their nutrients for the growing embryo within it as starch. And then once those seeds start to germinate, the starch is broken down into maltose. And that's exactly how beer is made and other malt liquors. they actually take whatever the seed is, whether it's hops or barley or rye or even corn. They start to germinate those seeds, so the seeds start to, grow a little bit. They add water to it, and so we humans harness the natural germination process, natural metabolism of these seeds to start breaking down their starch on their own into maltose, and then from there, alcohol brewers will mash. It's called the mash. They'll mash up the seeds and extract out, the excess parts to just get the maltose. Then yeast is added. We're going to come back to this when we get to respiration, cellular respiration, but the yeast can metabolize that maltose sugar and break it down into carbon dioxide and ethanol, drinking alcohol. And it's actually to make alcohol is we harness two different biological processes. So we harness the power of plants and the power of the fungus with the yeast to make that alcohol. Now, sucrose is obtained when a glucose and a fructose monosaccharides are covalently bonded together. You get sucrose. And this is what we call table sugar. So this is what you would bake with. Whether it's white sugar or brown sugar or powdered sugar, does not matter. It's all sucrose. And it's actually very chemically stable. It'll last pretty much forever as long as it doesn't get moldy or get bugs in it. Plants turn their products of photosynthesis into into sucrose because it stores so well and for so long term. And then our final one down here is lactose. If you bond a glucose and a galactose molecule together, you will get milk sugar. And this is unique, not to animals, it's unique to mammals only. So that is the defining characteristic of a mammal. It's an animal that produces milk to feed its young. It's not live birth because some mammals lay eggs. The platypus lays egg, but it does make milk. So that's what defines us as a mammal. Milk is actually modified sweat, but it's the sugar that's in it is lactose. And then most mammals stop expressing the gene for the enzyme lactase once they've gone through the weaning period and they're no longer needing that milk. But some humans have what we call lactase persistence, where we are still able to digest lactose even though we're adults, and that's how come we can drink milk or have dairy products. But this is still actually relatively uncommon across all humans. More people in the world cannot drink lactose. They're lactose intolerant. Our third category here are the polysaccharides. So these are the many sugars all covalently bonded together. The polysaccharides, organisms can use them for either energy storage or structural support. Okay, so for energy storage, you will need to know all of these names, okay, for your exams. For energy storage in plants, they use starch for their long-term energy storage. And starch, it has several different names, but the one that we work with the most in this course is amylose. So that's its proper name, amylose, and it's broken down with the enzyme amylase. So you can see this in, say, a potato. A potato is full of starch. The plant during the summer months will photosynthesize, convert all of those sugars that it makes into starch, amylose, and put it into the potato so the potatoes grow real big over the summer. And then in the winter, in places, not Florida that have winter, when, the plants die off, they're left with just their root system underground. The plant uses that stored up energy to stay alive over the winter and then also to regrow its stem and leaves in the spring. You can actually see this with a potato. If you take a potato and just throw it in the back of your cupboard or your pantry, if you leave it there long enough, it will sprout eyes, and if you leave it even longer, it'll sprout sprouts from those eyes, and you'll actually notice the potato part itself looks wrinkled and shrunken, shriveled, and it does that because literally the potato is digesting itself and then repurposing that energy to grow the stems. And when you look at amylose, It's basically you just bond glucose together covalently ad infinitum, and it will coil up for space purposes, but for the most part, amylose is just a straight chain of glucose. Amylopectin is a slightly alternate version of starch. It has a very few branches in it, but that one's less common. Animals, we use glycogen as our energy storage molecule, macromolecule. Glycogen is the same thing, it's the same monomer, so it's just glucoses, but they're all bonded in kind of the zigzaggy way. There's 1,4 linkages and 1,6 linkages is what they're called. There's more about it in your textbook. The glycogen is stored up in animals in the liver, if it's an animal that has a liver. And glycogen stores can last for a day or maybe two, depending on how many you had. Okay, so when you eat, your blood sugar raises, and then your liver pulls glucose out of your bloodstream, chains it up, covalently bonds it into a glycogen molecule, and then saves that for later when it's been a while since you've eaten. So that way your liver helps to regulate your blood sugar at a constant level. Now for structural support, this is mostly going to be found in your plants and your fungus. For animals, we have a different system, and we use proteins and extracellular matrix for our structural support. But for plants, they use a polysaccharide named cellulose. It's also called fiber. That's what we put it as on food labels. So cellulose is what plants use to make their cell walls. It's very similar to starch. It's just a bunch of glucoses chained together, but the linkages are different. So with starch, there's an alpha linkage, and with cellulose, there's a beta linkage. And if I zoom in here, you can kind of see the difference a little bit more clearly. So you can see on the starch, they have the hydroxyl groups here highlighted. They're all on the same side. And on the top of this molecule, you have that 6 carbon, at the top. But if you look at the cellulose, the glucose are bonded differently. And you see there's kind of an every other. So you've got a hydroxide down here, but then it's up here, then it's down here, then it's up here. Okay, it kind of zigzags. And the difference is immense. Most herbivores or omnivores do not have the cellular machinery to be able to digest cellulose. Very, very few. Your ruminants, termites, things like that can. But it's a pretty energy intensive process to digest cellulose. So we humans, we do need it in our diet. You do need fiber to help your digestive system and your bowels to function properly, and it helps reduce your risk for colon cancer. But we animals, for the most part, do not break down cellulose into glucose and then break that glucose down and turn it into ATP. We just don't have the enzymes for it. But a ruminant can, like a cow, but they have a 4-chambered stomach and they have to chew their cud, and it's a very long process for that to work. So it's a pretty strong molecule to make that cell wall for the plant. So, and here you go. I got a little bit ahead of myself, but herbivores pretty much, they only eat plants. So they have to digest the cellulose, but they don't have those enzymes. So they have different strategies to do that. So I didn't discuss termites yet. So termites actually have a symbiotic relationship with a little critter in their gut, and it's the little critter that actually digests the cellulose. It's a protist, which is a single-celled organism that is what makes the enzyme to digest cellulose. Ruminants, so this would be like cows, camels, llamas, giraffes. Okay, if it has hooves and it chews a cud, the ruminant. But they have a quite intricately evolved digestive system in order to be able to digest this. And they also have a symbiotic relationship with the bacteria in their digestive tract to do that. So their upper digestive tract is huge, basically. It's expanded, and it takes a long time to ferment, basically. The bacteria in their gut have to ferment the cellulose to be able to digest it down. And chewing their cud is maybe you didn't grow up on a farm, but I did. So a cow will eat the grass, it'll go in their stomach, and then they'll kind of burp it up, spit it back up into their mouth, and chew it again all day long. And that helps to digest that cellulose. And then there are also cacophores. These are, an example would be a rabbit or a hare. They don't have that expanded upper GI tract, the stomach. It's their lower GI tract, so the intestines that's expanded. So they don't regurgitate their food. They actually eat their feces, and they actually do look different if it's been through their digestive system once or twice. So they actually redigest their food, and it's the bacteria in their lower GI tract that helps to ferment these the cellulose, so they can extract nutrition from it. Okay, a few other carbohydrates. A polysaccharide, another polysaccharide is called chitin, the k sound. We see this in arthropods, which are insects, arachnids, and a few other things like millipedes or silverfish, things like that. Their exoskeletons use chitin, and also the fungus kingdom. The cell walls of any fungus are made of chitin. It's not strictly a pure polysaccharide, it's slightly modified. It has some nitrogen involved in it, but it's a different structural polysaccharide. So it helps protect the arthropods on the outside, and then the fungus cell walls, it gives them support. And then another polysaccharide is called peptidoglycan, and again, it's modified. So the peptide part refers to there's some amino acids and some protein parts, and then the glycan sounds like glycogen. That's the carbohydrate part. But bacterial cells use peptidoglycan in their cell walls. So it's just the animal cells that don't have the cell walls at all. So we don't have a structural carbohydrate. And that's the end of our section on carbohydrates.